So we're going to talk about specifically mitosis and then talk a little bit about meiosis and how they compare. So again, if you were in class or you've watched some of the other videos, we've already talked about mitosis a little bit. If your little sister, so let's pretend you have a little sister, if she came up and asked you, where is an example of a cell that goes through mitosis? You could say mitosis, and that's because mitosis happens in all of your body cells. Your somatic cells is what I want you to think of when you think of mitosis. So your muscles, your skin, your stomach cells, like those cells go through mitosis. And then I want to talk about the individual parts on the left here and draw the examples, and then talk about this pie chart, and that'll end our discussion about mitosis, and then we'll mention meiosis a little bit. So the first part about mitosis is going to be prophase, and then metaphase, and then anaphase, and telophase. But we're going to talk about the whole cycle as a whole, the cell cycle. So during interphase, and draw this example, you've got a cell, and you've got the nucleus. During this phase, again, what we mentioned before, 90% of the cell's life is spent in this phase. It grows during this phase. It copies its DNA during this phase. That would be S phase, and then it grows some more. This is where the cell does its job. Whatever job it's doing, this is where the cell does its job, is during interphase. Prophase is the first part of mitosis, which PMAT is the acronym we used to remember it. Prophase is where the nuclear envelope is dissolving because the DNA is too big to go through the pores. The nuclear envelope has to break down. So the example that I sometimes use is if we had built a car inside of the classroom, and then we were done building the car, we put it all together, but we can't drive the car out through the door or the window, we would have to break down some of the wall to get the car out. And that's what's happening. Here is the walls, the nucleus are being broken down. DNA is in the chromosome form. So if you'll notice, the DNA during interphase is in, looks like spaghetti. You're just kind of drawing a bunch of spaghetti looking stuff. Down in prophase, you're going to be drawing it more in, it's going to look like a chromosome, and it winds together. And then you also see these things on the ends, these two little green rods is how I drew it here, and they've got this black spiderweb looking thing that are starting to grow out. The green rods are called centrioles, and the spindle fiber is the little spiderwebs that are coming out, and they're starting to form. Then we get to metaphase. In metaphase, you should think middle, and that helps you remember that the chromosomes, the nucleus is already broken down, and the chromosomes are lining up in the middle. So the chromosomes line up, they're down the middle, M, and the spindle fiber is attached to each of these chromatids, and each of the chromosomes at the middle, and they're about to rip them apart. And what's beneficial about that is that this chromatid, sister chromatids, are exact copies of each other, because during interphase, when it copied the DNA, these two were in chromosome form. They were kind of stuck together. The left side is a copy, and the right side is a copy. And so then in anaphase, we see that the cell is going to start getting longer. This is you're going to start seeing the cell go through a little bit of cytokinesis. If you look at pictures online, it's not this simple. You see cytokinesis actually starts a little bit back here in anaphase and telophase. You don't have to know that, but just it is getting longer. The cell is getting longer. It's elongating, and the chromosomes are being pulled apart, and it looks like little A's. The chromosomes look like little A's. They are apart, being pulled apart or away from each other. So whatever anaphase helps you remember, apart, away, or the sister chromatids look like little A's or capital A's. And each of the sides needs to get the same chromosome. So these spindle fibers, it's important that they're strong enough to pull them apart to their side. We'll talk about later with meiosis, if one of these spindle fibers is not strong enough or something goes wrong, one side could get 4 and one side get 2, and then that could lead to problems. And let's go to telophase. Telophase, remember talking to a friend on the telephone, they're going to be very far away. And in this case, these nuclei are getting very far away. And we have this cleavage furrow that's growing or forming rather. And each of these nuclei are starting to form back. Their nuclear membranes, membranes are starting to form back, the nuclear envelope, and the chromosomes are starting to unwind back to what they looked like originally in interphase. But this is still one cell, separated nuclei. So this is nuclear division. These 4 stages, PMAT, that you see in a kind of reddish brown, they're all PMAT. These are all nuclear division. But then we see then the cell is still one. And then cytokinesis, which is the third step of the cell cycle. If you think about interphase being the first one, nuclear division is the second one, cytokinesis would be the third one. And you see 2 separate cells. This is not part of mitosis. This is when the cells split apart, and there are 2 daughter cells, and each daughter cell should be the exact copy of the parent. We can see that in a different form up here. We've drawn this already. You start off with one cell. You see these chromosomes. It goes into S phase, and so it's going to copy. DNA is copied. G phase is going to continue to grow. And then we get into PMAT, which is mitosis in this case. And then we get into cytokinesis. So it starts off as a cell with 2 chromosomes, ends up, this is still considered 2 chromosomes, but there's twice as much DNA here. So there's DNA here, and there's twice as much here, so that when you get over here and you split in cytokinesis, it's the same as over that we started with. And then each of these cells would go through the same process again and again and again and again. Just to clarify, G1, S, and G2 are interphase. PMAT, that's considered mitosis in this case. And then cytokinesis is the cell division. So growth, copying, going on here, nuclear division going on here, and cell division going on here. These are somatic cells. They're body cells. Somatic cells do mitosis. Every cell in your body, every living cell in your body, except for sex cells, so that would be a sperm cell or an egg cell, an ovum, those all go through mitosis. And just to kind of preview meiosis, meiosis is the same. It starts off the same. We've got a cell. It's a different cell, but the cells start off as a full cell, or we would call them a diploid cell. They start out the full amount, and then it goes through S phase, which copies, and it goes through PMAT, which ends up with 2 cells that look like the original. You see, that's how we ended up here. But down here on meiosis, it's going to go through PMAT and cytokinesis again. So what we end up with is cells here that are 2 of them, like we started off with, and then they get divided without going through S phase. So they divide again, which leads them to have half the amount of DNA that they started off with. But there's 4 cells that are there. So this would be a sperm cell or an egg cell. So that's this. The left side and this top is mitosis. This bottom right-hand side with the orange labeling is meiosis. I put it on here so you could see that it's very similar as far as the process goes. It's very similar to mitosis, but it has PMAT and cytokinesis added on here at the end. And in meiosis, we call this prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. So we call it, it's meiosis 1 and then meiosis 2.